This is our lab report number 3. It will be based on biochemical test part 1, which includes OF glucose medium, carbohydrate broth, and triple sugar iron agar test. This will be presented by Massiel Martinez, Mauricio Sanchez, and Juan Sanchez.
Introduction. The purpose of this lab was to gain an understanding of performing and interpreting the different biochemical tests that we utilize, such as the oxidative fermentation glucose test, the triple sugar iron agar test, TSIA, and the different carbohydrate broth tubes, which were glucose broth, lactose broth, and sucrose broth. The materials and methods for this lab. In this lab, we used the following cultures that are listed below. We also used the previously mentioned media of OF glucose, TSIA, and the carbohydrate broth tubes. And some of the other materials that we used were an inoculating loop and needle, and mineral oil, and a Bunsen burner.
Since there are 3 different types of media used in this experiment, each one had their own specific procedure. Beginning with OF glucose medium, you would inoculate the medium by taking a single bacteria colony using the inoculating needle and then stabbing the media about 1/4 of an inch at the bottom of the tube. One of the tubes would be labeled closed and covered with 1 to 2 milliliters of mineral oil. The second tube would be labeled open without any modifications. The caps on all tubes are to be left on loosely, and the bacterium was left to incubate for a couple of days. For the TSIA, the top of an isolated colony was touched by an inoculating needle, and then the TSIA tube was inoculated by stabbing the center of the medium all the way to the bottom of the tube and then striking on the surface of the agar slant. The caps were to be left on loosely, and the bacterium was left to incubate for a couple of days. For the carbohydrate tubes, the media was inoculated by taking a single bacterial colony using an inoculating loop, and it was inoculated carefully as we did not want to whack the glass of the Durham tube. The caps were left loosely, and the bacterium was left to incubate for a couple of days.
Observations. These are all of our observations for the different biochemical tests that we performed, or all of the media that we inoculated with bacteria. These observations were very important for interpretation, for concluding results and finding the characteristics of the bacteria. So here you can see our observations for the oxidative fermentation glucose test, the TSIA tests, and the different carbohydrate broth tube tests.
One of our first tests was the ONF glucose media. Here on the left, we perform several or 3 different kinds of inoculations with 3 different kinds of microorganisms, um, both having tubes with an open and a closed tube. Here on the left, we can see on the image of those tubes before being inoculated, being A. phasalis, P. Aeruginosa, and E. coli. So the principle of this ONF glucose media is to determine whether bacterium is oxidatively or fermentative using glucose. The purpose is to determine the atmospheric requirement of the bacterium. Bacteria that oxidize carbohydrates are usually obligate aerobes, whereas those that ferment are usually facultative anaerobes. So some of that biochemistry involved is as the organism grows and uses the sugar, the resulting acid would change the pH and the pH indicator would change colors. The indicator starts as that one on the left, as we can see, it's like a forest green color as a neutral pH, and then if it ends as a yellow with low pH and a blue with high pH. And these are results after. As we can begin with looking at A. phasalis, we can see a slightly different color change that can exhibit perhaps both oxidative and fermentative metabolism. So it might show some growth in both the tubes. But as for now, we can conclude that that it's not really a yellow, strong yellow, but it's just more of a green. So we can predict or conclude that there's no color change for both tubes. So the organism is asaccharolytic. Now looking at the middle test tube of P. aeruginosa, we can see that there is no color change. So we can conclude that it is asaccharolytic. Although P. aeruginosa is typically oxidative, it primarily uses oxidative metabolism when metabolizing glucose, so you would expect growth in the O tube with no significant acid or gas production in the F tube. Now on the very right, it is our E. coli. Here we can see there is a drastic color change as both tubes went from green to all yellow, but more with the closed tube. Therefore, it is a facultative anaerobe. Next, we performed the TSIA, a test on several different microorganisms, and the principle is to determine the ability of an organism to attack a specific carbohydrate incorporated in a basal growth media, with or without the production of gas, and along with the determination of possible hydrogen sulfide production. Now, here on the left, we can see the before of P. aeruginosa, E. coli, C. Furundi, and S. Typhimurium. And on the right, here are results after. Beginning with our P. aeruginosa, we can see that the slant is red, and this tells us that it does not ferment lactose or sucrose, so you would typically see that red on the slant. Now on the butt, we have a yellow butt. This tells us that it uses oxidative metabolism for glucose, so it may not ferment glucose or produce acid in the deep butt of the TSI medium, and therefore it results in that yellowish color. And we do not see that hydrogen sulfide production within P. aeruginosa. Now looking at our E. coli, both the slant and the butt are yellow, so they are both an acid-acid. This tells us that the bacterium is a facultative anaerobe, saccharolytic, and utilizes glucose, lactose, or sucrose, or even both. And we do see some slight gas production, and this occurs during fermentation, so that is why we see some gases. And we do not see that hydrogen sulfide production. Next, moving on to our C. Furundi, we see again that we have both a yellow slant and a yellow butt, telling us that it is an acid-acid. This bacterium is again also facultative anaerobe and a saccharolytic. The saccharolytic bacteria will utilize that glucose in the medium, producing acid byproducts, thus the whole tube will be yellow. And again, this is a capability of fermenting glucose, resulting in an acid. And lastly, for this slide, looking at our last tube on the very right with S. typhimurium, we also see a yellow slant and a yellow butt. S. typhimurium, looking at the slant, is capable of fermenting glucose, but does not ferment lactose or sucrose. So we would expect to see that yellow acidic slant due to that glucose fermentation. Now, similar to the slant, the butt would also produce a yellow acidic deep butt due to that glucose fermentation. So this bacterium is a facultative anaerobe, saccharolytic, and utilizes glucose. Now, looking at the rest of our microorganisms for the TSI agar exams on Staphylococcus aureus, Shigella flexurini, and Proteus mirabilis, we can see that before, then looking at the after results, we can see that all tubes turn yellow, meaning both the slant and the butt are acidic. So therefore, all of these three of these bacteriums are facultative, anaerobic, saccharolytic, and utilize those sugars. The saccharolytic, again, bacterium will utilize that glucose in the medium, producing acid byproducts, thus the whole tube will be yellow. Next, we perform the carbohydrate broth tubes using a glucose broth. On the left, we can see our before, with E. coli, P. aeruginosa, and Staphylococcus aureus. And to the right, we have our results of after a couple hours of inoculating those tubes. Beginning with our P. aeruginosa, we see no change. We see that there is a slight color change from orange to red, but this just means that that this is a negative carbohydrate reaction. The utilization of proteins in the medium increased the pH, causing the phenol red to change to a slightly darker red color, as we can see there. So from that, it uses that tells us that P. aeruginosa primarily uses oxidative metabolism for glucose, and it typically does not ferment glucose to produce acid or gas. And that is why it remains red, orange, and no gas production. Now, to the right of that, we have E. coli. Here we can see a big color change from that orange to a very yellowish color. This is this is as a result of a positive carbohydrate reaction, and this is because it is E. coli's ability to ferment glucose with the production of acid. In glucose broth, we can see that that color change went from red orange to that yellow due to that production of acid during glucose fermentation. And to the right of that, we also see for S. aureus that it also had a color change from an orange to that yellowish. It is a little less than E. coli, but it is still a result of a positive carbohydrate reaction. S. aureus is generally capable of fermenting glucose to produce acid. Now we performed our exam on lactose broth. On the left, we can see again how those tubes are very orange for E. coli and P. aeruginosa. Then to the right, after some inoculation of those microorganisms, beginning on our results for the after of E. coli, we can see that strong color change from that orange to that yellow. This is an indication of a positive carbohydrate reaction. E. coli is known for its ability to ferment lactose, which results in that production of acid. And for Puriginosa, we can see that there is slightly no color change. It's an orange to that little orange reddish, but this is indicating a negative carbohydrate reaction. Puriginosa typically does not fermentate lactose, and it is known as a non-lactose fermenter. So that's why we see a no significant color change and would remain red. Puriginosa does not produce acid during lactose fermentation or bubbles. Finishing up from with our results with the exact same carbohydrate broth too, but now we use sucrose broth. Here to the left, you can see how our tubes are very bright red, and this is before the inoculation of E. coli and P. aeruginosa. Now to the right, we have our results, beginning with E. coli. Here we can see a slight color change from that red, bright dark red to that slight orange. This can be an indicative of a positive carbohydrate reaction, although E. coli does have the capability to ferment sucrose, which results in the production of acid. This may be due to some interference, perhaps, or of poor or weak low count of microorganisms in that too. But it is supposed to turn yellow and be a positive carbohydrate reaction. Now, looking at our right tube of P. aeruginosa, we can see that that tube remains red, concluding that it is a negative carbohydrate reaction. P. aeruginosa does not ferment sucrose and is known as a non-sucrose fermenter. And that is why we see in that sucrose broth that there is no significant color change, and the phenol red just changed to a darker red color.
Our conclusions for the biochemical tests. For the OF glucose test, which helps to determine if the bacteria metabolize glucose fermentatively or oxidatively, we found that P. aeruginosa does not break down glucose because there was no color change, so it's asaccharidic. E. coli turned yellow, which means it's a facultative anaerobe and it does ferment glucose in the presence or in the absence of oxygen. And aphasalis, there was no color change, meaning it's also asaccharidic and does not ferment glucose. For the TSIA test, this test helps to determine the ability of the bacteria to attack a specific carbohydrate or carbohydrates. For P. aeruginosa, it turned red and there was also black precipitate. This tells us that it's a facultative anaerobe and saccharolytic, and that it also produces hydrogen sulfide, that it also produced hydrogen sulfide gas, and that it utilizes only glucose. And for E. coli, it turned all yellow. This tells us that it's a facultative anaerobe and that it's saccharolytic. So this bacteria utilizes glucose, lactose, and or sucrose, and it turned the medium yellow because it produced acid byproducts. And the rest of the bacteria here were also facultative anaerobes and saccharolytic, just like E. coli, according to our tests. For the carbohydrate broth tubes, the glucose tests, and these carbohydrate broth tube tests, help to determine the ability of the organism to ferment or break down these specific sugars with or without the production of gas. For glucose, E. coli turned yellow, meaning that it's a positive test. It does break down glucose. S. aureus was also yellow, a positive test. P. aeruginosa remained red, so this was a negative test for the glucose. For the lactose test, E. coli turned yellow, so this was a positive for the lactose carbohydrate. P. aeruginosa was red, this was negative. And finally, for the sucrose tests, E. coli and P. aeruginosa both were red, so they were both negative tests. Resources. These are two of the important resources that we used in this lab. We used the laboratory manual, pages 29 to 31, the lab journal, where we wrote all of our observations for the tests, and then interpreted these observations using the lab manual. This concludes our video lab report number 3 on biochemical tests part 1. Thank you for watching.